Hello and welcome to our session on serum and feeder-free differentiation of erythroid progenitor cells from hPSCs. My name is Selena Hallahan, Associate Product Manager at STEMCELL Technologies. In this presentation, I will give a brief description about novel methods for identifying treatments for sickle cell diseases. My colleague, Chau, will then discuss the use of PSC-derived red blood cells in research and describe some of the work that went into developing our new first-to-market product, the STEMDiff Erythroid kit. This new kit is designed for serum-free and feeder-free differentiation of pluripotent stem cells to erythroid progenitor cells that express glycophorin A and CD71. The STEMDiff Erythroid kit is a new addition to the STEMDiff product line, and I will touch briefly upon a select few products from this line before taking questions. In this section of the presentation, we will take a quick look at some of the research applications of red blood cells. Some of these applications may one day lead to novel disease treatments, and it's important to remember that no matter how far removed from the clinic you might feel, if you're researching erythroid cells, your work has the potential to someday directly impact patient treatment options. First, let's take a quick look at one of the more common hemoglobinopathies, sickle cell disease. This is a group of blood disorders that affects the hemoglobin molecule. The disease is caused by a recessive genetic mutation in hemoglobin that causes the red blood cells to develop a rigid, sickle-like shape, as can be seen on the bottom of the diagram of this slide. The abnormal shape of the cell can lead to a blockage in the blood flow and a shortage of oxygen to tissue. In addition, the sickled cells can break down prematurely and can cause anemia. As a quick refresher, here you can see a cartoon of a hemoglobin protein structure. There are hundreds of millions of these protein complexes in each of our 20 trillion red blood cells. The blue heme groups are the functional subunit of the hemoglobin that bind to oxygen, and the two globin chains, made up of alpha and beta subunits, are where the mutations commonly occur in hemoglobinopathies such as sickle cell disease. People with just one copy of a mutation here have sickle cell trait and are generally healthy. But those who inherit two mutant copies of this gene suffer lifelong consequences arising from the presence of this abnormal protein. Shortly after birth, babies usually stop producing fetal hemoglobin and switch over to producing the adult form of hemoglobin. However, some individuals continue to make high levels of fetal hemoglobin throughout their lives. This is referred to as hereditary persistence of fetal hemoglobin. Those rare individuals with sickle cell disease who also have hereditary persistence of fetal hemoglobin have an extremely mild version of sickle cell disease. As such, sickle cell disease is often treated using drugs that induce the production of fetal hemoglobin. For instance, hydroxyurea has been in use for the treatment of sickle cell since the 1980s and works by inducing the production of fetal hemoglobin. However, the patient's response to these drugs may diminish over time. To date, the only functional cure for sickle cell disease is a hematopoietic stem cell transplant. However, in addition to this being a difficult and intensive treatment for the patient, only about 10% of patients are able to find a suitably matched donor for the transplant. However, new technologies continually revitalize the field of medical research. In recent years, the rise of technologies such as CRISPR gene editing and pluripotent stem cell-derived cells have introduced new possibilities for understanding the biology of diseases, modeling diseases, and invigorating drug discovery for diseases such as sickle cell. These discoveries aren't going unnoticed. Looking at the currently active clinical trials published on clinicaltrials.gov, we can immediately see that there are three major themes in the clinical research for hemoglobinopathies. 1, drug development. 2, transplants and transfusion therapies. And 3, gene therapy. Indeed, we've already seen the clinical potential of gene therapy for sickle cell disease. In 2017, a group in France reported the first successful treatment of a patient using gene therapy. The group modified the patient's own hematopoietic stem cells ex vivo with a healthy hemoglobin variant. This was done by lentiviral transfer of a modified beta globin that encoded an anti-sickling variant. The modified cells were then successfully transplanted back into the patient. After more than 15 months following the transplantation, only minor side effects from the treatment were reported, and there were no reported sickle cell disease-related clinical events. Even more recently, a group at the Sarah Cannon Center for the Blood Cancer in the United States published initial results from their work in January of this year. Their approach uses CRISPR gene editing to increase the levels of fetal hemoglobin in the red blood cells by turning off BCL11A, a silencer gene for fetal hemoglobin. Over a year after treatment, the 2 patients treated with the modified cells showed high levels of edited cells in both the blood and bone marrow, as well as increases in their fetal hemoglobin. They also showed transfusion independence and the elimination of vaso-occlusive episodes. So, say you're setting out to study sickle cell disease and to develop a new treatment using gene editing or novel drugs. Where do you start? Well, first you have to choose what cell source you'll be working with. The most obvious choice is doubtless primary cells, sourced directly from patients and/or healthy donors. However, there are challenges with these cells. For example, primary cells are difficult to maintain in culture. Donor-derived cells are inherently variable between batches, and of course, it is way more difficult to obtain disease-specific samples, as these require donation from patients who are already ill, meaning that disease samples are inherently more precious. Some groups may use immortalized cell lines instead. The same considerations remain here as they do for any group using immortalized cell lines. For example, karyotypic instability and a lack of diversity of disease phenotype. A newer option in comparison is to use PSC-derived cells. So, why choose the relatively new option of PSC-derived cells? Your decision might be influenced, in part, due to the fact that it takes a lot of money and time to develop new treatments. The drug development pipeline is incredibly costly and long, partly due to the high attrition rate of compounds as you pass through each phase of development and eventually make your way into clinical trials. We already know that cells can be used to help predict what might happen when these novel drugs get into a patient. Cell-based models can be used in an effort to narrow down key compounds earlier on in the drug development pipeline, or more realistically, to exclude those that might cause issues when they get to the clinic. And while mature blood cells are usually very easy to access, the challenges discussed in the last slide, the difficulty of accessing disease samples, difficulty in maintaining primary cells in culture, and donor-derived cell viability remain. As such, hPSC-derived models can be an attractive option for disease modeling. They remove a number of these obstacles, as well as allowing the researcher to create unique mutations and standardize their screening protocols with fewer variables and more cells to repeat experiments. So these are some reasons why you might want to use PSC-derived cells in general. I'll hand it over now to Crystal to discuss the use of PSC-derived red blood cells in your research.
Thank you, Selena. In this section, I will go over some background information on hPSC-derived red blood cells. Over 100 million units of whole blood are collected worldwide every year, and with the progressive aging of the population, it puts a strain on blood donors. With human pluripotent stem cells, they can provide an inexhaustible source of erythroid cells that can be used in studying developmental biology, drug discovery, disease modeling, cell replacement therapy, and gene therapy. Because of these applications, the robust and efficient generation of clinical-grade erythroid cells from pluripotent stem cells is an area of growing interest. Although it is now more routine to derive patient-specific iPSC lines, differentiation of these pluripotent stem cells into the cell of interest still remains a challenge. For the success of hPSC-derived cell applications, we need to know what cells we need and how to differentiate them to that cell type. The biggest hurdle in the field is the generation of adult-like red blood cells with high yields and efficiencies, which is required for disease modeling and drug discovery. So far, the development of human erythroid cells has mainly been studied in models of adult hematopoiesis. Therefore, regulatory processes involved in the specification of hematopoietic stem cells during embryogenesis remains not well understood. Hematopoiesis takes place in distinct waves, with each wave producing cells that increase in complexity of blood lineage potential. Erythroid cells are generated in 3 main waves throughout developmental hematopoiesis. These are highlighted by the green boxes. In the first wave, the yolk sac produces primitive erythroid progenitors, which in turn produces large nucleated red blood cells that express embryonic hemoglobin molecules. These are known as the primitive erythroblasts. In the second stage, erythroid progenitors emerge from the vascular hemogenic endothelium in the blood island regions of the yolk sac. They then dislodge and enter systemic circulation and seed the fetal liver. This is considered definitive because these erythroid progenitors differentiate into blood cells with adult-type features. In the third wave, hematopoietic stem cells arise from the hemogenic endothelium in the AGM. These are the definitive cells that are more engraftable. So we know that erythroid cells are generated in both the primitive and definitive waves of hematopoiesis. Now looking at this figure, you can see that primitive erythroid cells express embryonic and fetal globins and lack the expression of adult globin. In the second wave, the erythroid cells express a combination of embryonic, fetal, and adult globins. And then the hematopoietic stem cell-derived definitive erythroid cells express adult globins. So this is looking at gamma or beta globin, and they lack the expression of embryonic globin. In the table on the right, the key differences between primitive and adult-like erythroid cells are highlighted. The first trait is globin expression, and primitive erythroid cells express embryonic and fetal hemoglobin, while definitive erythroid cells express fetal and adult globin. The next trait is enucleation capability. There is none to very little enucleation in the primitive cells, while in definitive cells, the majority of cells are enucleated. Then looking at cell size, primitive cells are larger, while definitive cells are smaller. It is also important to note here that there are no well-established surface markers that distinguish primitive and definitive cells. The use of hPSCs in the generation of mature red blood cells is a fast-developing field with many published protocols. But this field is still growing. None of the existing protocols are well established, and many protocols rely on the use of feeder cells or serum as part of the protocol. There are some key challenges in the generation of hPSC-derived erythroid cells. First of all, the cells are highly sensitive and fragile, and in the field, it has been difficult to generate adult-like red blood cells from pluripotent stem cells. These difficulties include the inefficiency of cell maturation and enucleation, as well as very little beta globin expression. There's also a lack of robust protocols that can differentiate hPSCs to erythroid cells with high yields and efficiency. So the generation of erythroid cells from PSCs consists of 3 main phases. The first one being hematopoietic specification, and this is where the pluripotent stem cells are specified towards a hematopoietic progenitor cell. The next one is erythroid differentiation, and this is where hematopoietic progenitor cells are differentiated to erythroblasts. And the last one is erythroid maturation, where the erythroblasts further mature into red blood cells. So with these challenges in mind, we have developed a kit that addresses the first 2 phases. So in this next section, I'll introduce you to the STEM-Diff Erythroid Kit, which was recently launched. The STEM-Diff Erythroid Kit comprises of serum-free and feeder-free media and supplements used for the differentiation of human pluripotent stem cells to CD71 positive and glycophorin A positive erythroblasts. This is a 2D protocol performed in 2 stages. During the first hematopoietic stage, H phase, cells are induced into hematopoietic progenitor cells and harvested from the culture supernatant. Cells are then further differentiated to erythroid progenitor cells in the erythroid phase, E phase. Following this, there is an optional protocol for maturing cells further into normoblasts and reticulocytes using EPO and human AB serum. The STEMDiff erythroid kit provides high yields and efficiency of erythroid differentiation. Now we will go a little bit more into detail into the two-stage protocol. The first stage is a hematopoietic specification of human embryonic stem or induced pluripotent stem cells. At day -1, the ES or iPS cell aggregates are seeded on matrigel-coated plates. Then at day 0, the number of attached colonies are counted, and the wells with 4 to 10 colonies are selected for a full medium change to medium A. This is followed with a half medium change at day 2. On day 3, the medium is changed to medium E1, and the full medium changes are done at day 5 and day 7 before harvesting the hematopoietic progenitor cells in the supernatant at day 10. You can see in this flow plot on the right that the harvested cells express CD34 and CD43, which is what we expect with these hematopoietic progenitor cells. At this point in the protocol, you do have an option to cryopreserve the cells to give you more flexibility in scheduling. Once you are ready for the erythroid differentiation stage, their hematopoietic progenitor cells are then seeded into medium E2 at 40,000 cells per mil. The second stage lasts 14 days with supplement replenishments every 2 to 3 days and a passage time point at day 17. At day 24, the erythroblasts are harvested and the cells are highly expressing glycophorin A and CD71, which are markers of erythroid cells. You also have the option of cryopreserving these cells at day 24 for further downstream applications. Now, looking at the performance of the STEMDiff erythroid kit, in Figure A, the average numbers of viable glycophorin A positive erythroid cells generated by day 24 per input PSC is shown. We have 3 ES cell lines, H1, H7, H9, and 2 iPS cell lines, 1C and F016. Across all 5 cell lines, over 10,000 Gly-A positive cells are produced per input PSC. In Figure B on the right, the overall frequency of glycophorin A positive erythroid cells after 24 days is shown as a percentage of the total live cells present. On average, over 75% of the cells are glycophorin A positive. To further characterize these cells, we looked at hemoglobin expression at day 24. The data shown here is qPCR data for 2 different cell lines, 1C and iPS cell line, and H1 and ES cell line. For both cell lines, erythroid cells generated with the STEMDiff erythroid kit express a mix of primitive and definitive hemoglobin. In Figure B, we show a picture of the cell pellet at day 24, and we can clearly see that the cells are well hemoglobinized. And then in Figure C, we show a cytospin image at 40 times magnification of day 24 cells stained with May-Grunwald-Gimza stain. These cells display typical erythroblast morphology with some large basophilic erythroblasts and some smaller, more differentiated normoblasts. So earlier I talked about how the STEMDiff erythroid kit is a 2-step protocol, but you can also perform an optional erythroid maturation step with the cells harvested at day 24. Cells generated from the STEMDiff erythroid kit can be plated at half to 1 million cells per mil in STEMSpan SPEM2 with 3% human serum and 3 units per mil erythropoietin. This is a common maturation method used in the field, and after 3 to 4 days, there is a medium top-up, and then at day 7, the cells can be harvested. Further maturation of the erythroid cells results in smaller cells with condensed nuclei, which is shown in Figure A. This is typical for orthochromatic normoblasts. In Figure B, a representative flow cytometry plot shows that over 90% of the cells are highly expressing glycophorin A. A majority of the cells have decreased CD71 expression, which is consistent with erythroid maturation. In Figure C, this bar graph summarizes the average frequencies of glycophorin A expressing erythroid cells after the maturation culture. For all cell lines, there was above 90% of glycophorin A positive cells. And then in figure D, we show the fold expansion within the 7-day maturation phase. Notably, this fold expansion data shows that the cell numbers are maintained during the maturation culture. We do not expect to see expansion of cells during the maturation phase, but what we do want to see is a maintenance of cells, which is what we see here. Finally, I just want to give a quick summary of the STEMDiff Erythroid kit. This kit is a two-step serum and feeder-free system for the differentiation of hPSCs to erythroid cells. It only consists of 2 basal media and 3 supplements, which provides a simple, straightforward feeding schedule. Over 75% of cells generated are glycophorin A positive, and 10,000 erythroid cells are generated per hPSC. These erythroid cells are well hemoglobinized and express all 3 hemoglobin types. The PSC-derived erythroid cells can then further mature into orthochromatic normoblasts with over 90% glycophorin A positive cells and maintenance of cell numbers. Overall, with the STEMDiff Erythroid kit, we can generate erythroid cells from hPSCs with high yields and efficiency. If there are any other questions about the STEMDiff Erythroid kit or about any hPSC-derived red blood cells, there will be time at the end of the presentation for questions. Now I will pass it back to Selena, who will be speaking about some of our other STEMDiff kits.
Thank you, Crystal. I will now briefly introduce you to some of the other STEMDiff kits in our portfolio, which may be of interest to hematopoietic researchers. As the pace picks up in research around therapies for hemoglobinopathies and other diseases, it is important to ensure that you are testing novel compounds and editing conditions on relevant cell-based models. hPSC-based models hold tremendous potential for the study of human development and disease. Stem cell technologies is ideally situated to support the generation of clinically relevant PSC-derived cell models with simple and effective differentiation kits and maintenance media that is suitable for the clinic. In addition to being one of the most widely published medium for hPSC culture with over 1,500 peer-reviewed publications, teaser culture is now manufactured under relevant cGMPs, ensuring the highest quality and consistency for reproducible results. Find out more about how STEMCELL can support your regulatory needs at stemcell.com/compliance. However, when you're working with hPSCs, even the most detailed and rigorously followed protocols can still lead to inconsistent differentiation. The STEMDiff family of products have been developed to promote reproducibility in your research through careful optimization of media formulations, raw material specifications, and manufacturing processes to minimize reagent variability. In addition, STEMDiff products include detailed, user-friendly protocols to standardize your differentiation procedures. In addition to those outlined on this slide, there are STEMDiff kits available for differentiating PSCs to all 3 germ layers. You can find out more about the STEMDiff product portfolio at stemdiff.com. The first kit we'll discuss is the STEMDiff T-cell kit. This is a relatively new addition to our STEMDiff portfolio, launching just last year. This kit is serum and feeder-free and differentiates PSCs into CD4, CD8 double positive T cells. The protocol for the T-cell kit takes place in 2 distinct sections. Firstly, PSCs are differentiated to embroid bodies, and CD34 cell positive hematopoietic cells are isolated. They are then further differentiated, and double positive T cells can be harvested after 28 days. Cells were harvested and analyzed for the expression of CD3, CD4, CD8, and TCRA alpha by flow cytometry. This slide shows representative flow cytometry plots that indicate the successful generation of double positive T cells from PSC-derived CD34 positive cells after 28 days of culture using the STEMDiff T-cell kit. Figures A and B show T cells differentiated from embryonic stem cells, and figures C and D were differentiated from induced pluripotent stem cells. Also launched last year, the STEMDiff NK cell kit is serum and feeder-free and is optimized for differentiating PSCs into natural killer cells that express CD56. Similar to the T-cell kit, the protocol for generating NK cells using this kit takes place in two sections. Like the T-cell kit, cells are first differentiated into CD34 positive cells in a 12-day protocol. Following this, they are further differentiated into NK cells, which can be harvested after 28 days of culture. Here we can see representative flow cytometry plots for some of the common NK markers. All cells show the expression of CD56. In addition, figures A and B show the expression of NKp46 and NKp44, respectively. Figures C and D show the expression of NKp30 and NKG2D. And in figures E and F, we can see expression of KIR and CD45. These plots were analyzed following 28 days of culture with the STEMDiff NK cell kit. The final kit that we will discuss today is the STEMDiff monocyte kit, which is also serum and feeder-free. Unlike the T and NK cell kits, the STEMDiff monocyte kit uses a 2D protocol to differentiate PSC cells to monocytes. CD14 positive monocytes can be harvested directly from the culture supernatant starting as early as day 14, and these can be repeatedly harvested during the rest of the culture period. The STEMDiff monocyte kit produces CD14 positive monocytes from hPSC-derived cells, as seen in figures A and B on this slide, and ES-derived cells, as seen in figures C and D. Monocytes generated using the STEMDiff monocyte kit can also be differentiated into M1 and M2 macrophages, or into dendritic cells, depending on the culture conditions that are used. In summary, PSC-derived cell models hold tremendous potential for progressing your research, whether you're researching the basic biology of a cell or investigating a novel compound for the pharmaceutical treatment of disease. M-teaser culture media is now manufactured under relevant CGMPs, preparing your work for the next step. And the STEMDiff portfolio, when combined with M-teaser culture media, supports a standardized PSC workflow. Learn more about how STEMCELL is positioned to help your PSC and hematology research at stemcell.com. We will now take questions from the audience.
Hi, everyone. Thank you for joining us for this wonderful talk today. My name is Mina, and I'm the moderator for the Q&A session this morning. I'm just gonna go ahead and ask a few questions that have come up in the Q&A session. Please keep asking your questions in the Q&A session. We'll be able to see them and answer them as we go on. All right, Selena and Crystal, just the first question for you is, what is the best maintenance media to use with the Erythroid kit?
I can answer that. So maintenance media, there's quite a few that you can use. You can use M-teaser, M-teaser Plus, and teaser E8, all from STEMCELL, and they're all readily available, very easy to use.
Thanks, Crystal. For the second question that we have, they're asking if you have ever tried using blood cell-derived PSCs as a starting source for this kit.
Yes, so I have tried using blood cell-derived PSCs, and they work just as well as any other cell line. So definitely works with blood cell-derived.
Thank you very much. A question that we have received in the past for this kit has been, do cells generated using the Erythroid kit produce colonies in the CFU assay?
Answer is yes, they do generate CFU. So they generate both myeloid and erythroid colonies. So CFU-G, MGM for myeloid, and also produces BFU-E and CFU-E colonies.
Perfect, thank you. Can you also maybe comment on the cryopreservation media, if there's any specific freezing medium that you recommend for the PSC-derived erythroids?
Yes, so the cells from the STEMDiff erythroid kit can be frozen down at either day 10 or day 24 of the culture, just to make your workflow a little easier and more flexibility. And it can be frozen down in quite a few different media. So one of them is Freezer S, and then there's also CryoStor 10, And you can also freeze it in 10% DMSO in FBS. My favorite is Freezer S. It has the best viability and recovery, and the cells work amazing afterwards.
Awesome, thank you. One thing that I think many people are curious about is the gene editing aspect for these cells. Can you maybe tell us if cells that you derive using this kit can be genetically modified in vitro?
I personally have not tested gene editing on these cells, but I don't see any reason for it to not work. It should work just as well as any other cells that you derive.
Perfect, thanks. And I think that's all the questions that we have. Do you, Selena or Crystal, have any comments you would like to further expand on for this?
I was just going to mention that we do have an exhibitor booth in the exhibit hall here, so you can go there to find out a little bit more about the STEMCELL Erythroid kit. We've also got some information about our hematopoietic kit and some more information also about those maintenance media, including the teaser line of products, which, as I mentioned in the presentation, are now manufactured under CGMP. So if you'd like to know anything more about that, we have some of our staff at our exhibit booth in the exhibit hall.
Thank you, Selena. Crystal, thanks for the talk and the Q&A. I think that's all that we have for today. I don't see any more questions coming in. I think we are good to go for now. Thanks everyone for listening in.
Thank you.
Thank you.